Thank you, Chris. Yeah, so I'm not going to talk about consciousness, nor will I speak about evolution. I don't have access to Neanderthal neurons. I wish I would. So is the screen correct, Jonathan?
Yes,
is this all good? Just give me a thumb.
Yep.
Okay,
we're good to go.
All right. Yeah. So this is a 7-year. This is the outcome of a 7-year study that we did at the Allen Institute, involving local area surgeons, together with a large team at the Allen Institute, together with many collaborators. Patrick Hoff, Rafa Juste, Idan Seger, Christof Koch, Hype Mansfelder, Gabor Tamas, and others. So it's, the paper has like 140 co-authors, and it's primarily funded by the Allen, but co-funded by the Allen Institute. We know since roughly 200 years that all biological organisms consist of one or more cells. And then, of course, Raymond Cajal and Golgi showed us the variety of nerve cells, the dizzying variety of different neuronal morphologies. And over the last 30 years, we've recognized that there aren't just 2 or 3 or 4 or 5 or a dozen, but there may be several thousand types of brain cells, neurons and non-neurons, in a typical brain, whether they're certainly of all animals that we've investigated, which of course gives rise to the question, what's all the function that I won't talk about. When I came to the Allen Institute in 2011, I had worked very successfully with Itzhak Fried, a neurosurgeon, and we've recorded over many years in patients, in epileptic patients, single neurons, things like the Jennifer Aniston neurons. So neurons that are very specific, that fire in response to specific, very familiar individuals, politicians, celebrities, or loved ones that the patient was familiar with. And we found that many neurons in these patients that people otherwise might think of as sort of near diseased or near pathological brains were highly, highly functional. And so I was very interested to start a program at the Allen Institute that would focus on the physical, biophysical, transcriptional, and morphological properties of these neurons. In particular, then a paper appeared by Gabor Tamas out of Szeged University in Hungary that's done exactly that took neurosurgical material and recorded successessly from individual neurons in slides. So this is all slides. None of this is in vivo, it's all in vitro. So think of this like a comatose brain. But it's a living brain. So, and we know, of course, that the neocortex is disproportionately expanded in human compared to the mouse or other mammals, both in its volume relative to subcortical structures, and particularly the proportion occupied by the supragranular layers that selectively make connection with other cortical and telencephalic structures has been greatly expanded. And single-cell transcriptomic analysis of human and mouse neocortex and also non-human primates show an increased diversity of glutamatergic neuron types. So to probe this functional and anatomical correlates of this transcriptional diversity, we set up a robust patch-seq platform where we do 3 things in the same neuron, in the same nerve cells. We insert a patch electrode. We do a brief electrophysiological profile, 5 to 10 minutes, to characterize the electrophysiological properties, step pulses, ramps, current, white noise, frozen white noise. We inject biocytin for subsequent recovery and reconstruction of the dendrites and the proximal axons. And then we suck out the nucleus in order to obtain reliable transcriptional mRNA information. We've done this year in roughly 90 donors from 3 different local hospitals. So we've worked over the last years to set up this dense network of neurosurgeons. And we've had sort of progressively, we started this, I think our first ever recording was in 2014 in January. And certainly before the pandemic hit, we had on pretty much 1 sample per week. of course, it's a random process. So sometimes we have 3 samples in 1 week, and then let's say in January, we may have no sample whatsoever. But typically, we have like 50 samples a year. Each sample is roughly the volume of a sugar cube, roughly a cubic centimeters. They come from both. They derive from, the majority comes from epilepsy tissue. So this is where the surgeon has identified a single focus or suspected single focus. And then in order to access a deep tissue tumor, let's say, or epileptic foci, the surgeon has to cut through the overlying middle temporal gyrus. Usually, most of the tissue is from middle temporal gyrus. 20% is also from frontal areas. Very rarely is it from the back area. And so we get tissue that's distal from the site of the tumor or distal from the site of epileptic seizure. We have a rapid transport system to move it. We have an oxygenated chamber, temperature controlled, etc. in a car. We drive it quickly to the Allen Institute, and typically within, I don't know, an hour or so, it's in our lab. And now we dissect it, we cut it, we slice it, we divot it out because lots of people want access to this tissue for a variety of different processes. And so typically, within 2 hours, we can start recording from this tissue. And of course, this tissue is, 2 hours ago belonged in someone's brain, and you might be recording from the neurons that's involved in their memory of the first kiss or whatever, right? So this is human tissue. What we've discovered and published about extensively, unlike mouse tissue, that typically after 12 or 18 hours, you can't really, the gradients run down. You can't really record high quality data anymore. In the human tissue, possibly because human tissue, of course, has to live much longer, we can routinely record and get beautiful synaptic potential, action potential, fast firing interneurons, the most fragile ones, 72 hours after the tissue is resected. So it's really it's a high quality. We've never found, we've done detailed statistical analysis. All of this data, by the way, is, of course, like always with all of our materials on our website. You can download all the data, all the metadata, all the individual neurons. So we've done a lot of analysis, to look at things like electrophysiological properties to see that they aren't really statistically different between, let's say, epileptic tissue and tumor tissue in terms of resting potential, action potential, height, initiation, FI slope, et cetera, et cetera. I mean, it has been a consistent critique of these studies that there must be large variation associated with disease state and neuropathology that sort of, obscures any coherent result. But based on my early results, with in vivo, with patient recording, I was slightly skeptical. And our experience, and now buttressed by a lot of statistics, shows that we can get highly consistent results using neocortical tissue that's distant to the site of the orbis pathology. So for this study, we used histological markers to evaluate, let's say, NISL, to evaluate cellularity, neuronal density and layer orientation, astrogliosis, activation state of microglia. We looked at non-phosphorylated neurofilament age using, SMI32, and we looked at cellular proliferation. And all these immunostained tissues for each donor of these 90 donors, all this tissue were digitized and independently and blindly scored by 3 neuropathologists with a number score between 0 and 3. And so we've done this statistical evaluation. We found really very little evidence for consistent disease or pathology-related alteration of the physiological features measured when using cortical tissue from the middle temporal gyrus or frontal cortex. So indeed, what I think of is remarkable result from our study is a stereotypy of neuron cell types measured transcriptomically, morphologically, and physiologically across human, across 90 different tissue donors with many uncontrolled axes of variation, right? There's age, gender, ethnicity, disease condition, severity, et cetera, But the basic cellular print seems to be highly robust across all these 90 patients. All right, so we do, as most of you all know, we have since many, many years this pipeline where we do both single cell and single nucleus RNA sequencing. It's a beautiful technique. It scales up very well to now millions of cells. So we can, so we do all the steps of RNA processing and sequencing as for mouse patch-seq cells. So we have this large pipeline that can be fed either by mouse tissue or by human tissue or non-human primate or any other tissue. The main difference between the human and the mouse is for the human, we use only nuclear RNA using the SmartSeq V4 ultra-low input RNA kit from Takara to reverse transcript polyRNA and amplify, full-length cDNA. And typically, each sample was sequenced to approximately a million read. And we've done comparison in the mouse between, cytoplasmic and nuclear RNA, and we understand well that relationship. Now, what you see which to me was surprising. So the last common ancestor, between mice and us roughly lived by the time the asteroid came in 60 plus million years ago and wiped out much, if not most of life on planet. Despite that sort of 60 million years, we can really very nicely find cell types aligned, not necessarily at the it's a hierarchy tree. We're doing this taxonomic analysis of transcriptional defined cell types. This was in a paper that came out 2 years ago. And, where we roughly find, give or take, 50 excitatory glutamatergic cell types in each brain area and roughly 50 GABAergic cell types in each area. And we can find really, in some cases, even down at the very bottom of the taxonomic trees, we can find like Schwann-Lillet cells. There's one type of Schwann-Lillet cells in the mouse, and there's one type in the human. But here you can see for excitatory, so here we're just focusing on excitatory cells. You can see on the x-axis, we cluster either from mouse primary visual cortex on the left, or on the right, we cluster a secondary motor area, ALM. And we cluster that against human data. This is human data that's either from postmortem, so that was in an earlier paper, either postmortem or neurosurgery, by and largely cluster along the diagonal. So there is corresponding cell types. It is not true, as many people have surmised, that there are many more cell types in humans than in mouse. The number is roughly the same. It's surprising to many, because many people had expected, obviously, given our different cognitive ability, there to be many more cell types in cortex, particularly in human than in mouse. It's not the case. It's roughly the same number. And you can match many. And if you look at the upper left-hand corner of this matrix, you can see there are 3 superficial cells that we find, excitatory glutamatergic cells that we find in layer 2-3 in the mouse, that matches collectively, not individually, but collectively 3 layer 2 types in the humans. And we call them, we call each cell type, we have to give them some name, these beautiful names that are very evocative, like LTK or GLPR2R or FREM3. We call them after sort of one of the dominant genes that's expressed. It's not the only gene, of course. Typically, there are 30, 40, 50, 60 different genes that characterize each cluster, that are highly expressed in each cluster, but we name them after one. So we have this one cluster where we have 3 mouse glutamatergic upper layer cell type that matches onto 3 human. And then we find 2 new human cell types, CAR M1P1 and called 22A1, that are only found in deep L3. So these seem to be different from the mouse. In the mouse, interestingly, they correspond to layer 5 cells. Like somehow there was a shift from layer 5 up into layer 2-3 in the human across, the 65 million years of common evolution. So though all 5 human T-types mapped to the intratelencephalic mouse subclass. So transcriptional, all of these 5 human cell types seem to map onto the mouse intratelencephalic, in other words, the primarily cortical projection, consistent with supragranular neocortex, solely being composed of such cortical, and telencephalic projecting neurons. Surprisingly, these 2 deep layer 3 human cell types are more similar transcriptionally to infragranular L5 and L6 IT types in the mouse. And this result seems to generalize across V1 and ALM. All right, so there are 3 matching types in human and mouse, and 2 new ones in the human that are supragranular that in the mouse are infragranular. All right, so what PatchSeq, as I mentioned, allows us to do, we can do all 3. We can match the electrophysiology onto the anatomy, onto the transcriptional identity of the cell. So we can see how M, L, and MT and E type map onto each other. So we primarily, as I said, sample human middle temporal gyrus and frontal areas, is particular anterior cingulate cortex, since that's where most of the neurosurgical sample come from. And so we compare this. I mean, we're just driven by the tissue we get. So we compare this material to mouse V1 and mouse temporal association area (TEA), which is often used in comparison with human MTG. We don't really find big differences. So the combined thickness, it's a little bit more than 1 millimeter, of the relatively thin layer 2 and very thick layer 3 in humans, so indicated there on the left, is a little bit thicker than all of mouse cortex, right? So clearly, mouse cortex, is on the order of 1 millimeter, and in us, it's between 2.5 and 3 millimeter. So there we use new N labeling. We found that the density of human neurons, is in this case 28,000 cells per cubic millimeter, with a high density in layer 2 that decreases systematically to a low point in mid layer 3 of about half that. So have much higher density, up to 150 or 160,000 cells per cubic millimeter. But of course, they're much bigger in human, these cells, and they're expanded. So the approximate number is roughly the same under 1 square millimeter of cortex, roughly. L3 is often divided here into 3A, 3B, and 3C. We don't see sharp changes in cell density or soma size. But we do see the cells, as I said, increasing, corresponding to the lower density in humans. These cell increase in cell area until they really get very large, sort of in the midpoint of layer 3. So in the middle diagram, you can see we did UMAP, this dimensional reduction onto 2 dimensions using UMAP, and you can see this sock-like shape, right? Shaped like a sock, and we can map all these 5 glutamatergic cells. You can see they map onto this continuum. So where the distance in this UMAP projection among cells approximate overall differences in gene expression. Here it's roughly 2,000 genes, and cells so from the same T-type grouped together. So the overall distribution for humans forms an extended continuum across the LTK, GLP2R, FREM3, and continuing to the CAR M1, P1, and COL22A1 cell types. They're sort of located on a separate island. While the similar analysis of layer 2-3 mouse types show much more compact distribution in both V1 and the lower right, you can see. Well, on the right, you can see mouse material from either V1 or TEA, and it's much more compact in this UMAP representation. So interesting, the largest T time, the FREM3, as you can show in the middle plot on the right, really shows a particular extended graded distribution that varies as a function of cortical depth. I'll come back to that. So here you can see where we plot the location. So these 5 cells seem to be systematically expressed at different locations, right? Just using the cell body in the initial material as a marker from the surface of cortex, going from the left to the right to deep layer 3. This is all just layer 2 and layer 3. And you can see on the right, we did this human MFISH using multiplex fish, and you can the red dots are the cell types, the T types using those 5 different cell types, LTK on the left till all the way over on the right called 22A1. So you can see the systematic shift from cells that are expressed just at the top of layer 2, just at the boundary with layer 1, all the way to the upper part of layer 4. Scale bar there on the lower right is 100 microns. And then we do this morphology reconstruction, right? So this is biocytin, and we have Stacy Sorensen. She runs this anatomy department here, the anatomy pipeline, where we do this routinely. So as I said, one of the most obvious differences is the cell size and the location of the soma and the overall length of the sort of dendrite. And so you can see that transcriptional defined LTKs is really relatively short. They're primarily in layer 2 and upper layer 3. They exhibit, you can see below, the electrophysiological properties. They exhibit a very regular firing pattern with little firing rate adaptation and no sag in response to current steps. Morphologically, they're short, extending multiple apical dendrites into L1. So they would sample the feedback that goes into layer 1. GLP2R neurons are primarily in upper layer 3. They lack firing rate adaptation. They have high input resistance and a pronounced sag. They also have fewer dendritic branches for their longer apical extent, and often have a distinct apical tuft in L1. So that's the third group. The second group from the left, those are the FREM3, and in our hands, it's the majority of cells. We have like 360 something neurons here, and the FREM3 T-type, they represent 57% of all our neurons. And it's interesting, you can see the laminar distribution spanning the distribution of both LTK and GLP2R with morphoelectrical properties that overlap but are distinct from those 2 types. And you can see this beautiful gradation, right, of those neurons from the ones that are just located in the upper part of layer 2 have almost no apical dendrites, and the ones that are located sort of towards the bottom of layer 3 have this very long apical dendrites, like they're pinned to the top there. So the two deep L3 types, layer 3 types, CAR-M1P1 and COL22A1, they have distinctly different dendritic properties. The CAR-M1P1, they have distinct apical obliques and basal dendrites branching near the soma, as well as they have fast action potential upstroke, if you look at the phase diagram representation of individual action potentials. And then the ones on the extreme right, COL22A1, have very sparse basal and apical dendrite. Sometimes they effectively have no apical tuft. They have very steep firing frequency to current steps, partly because of the very high input resistance. We also reconstructed the axon of a subset of these neurons, not all, because that's very laborious given the extensive local axonal tree. Of course, because these slices are 350 micrometers thick, we don't recover anything outside those 350 micrometers. But within that, the greatest local axon innervation surrounds the soma in L2 and L3, which is really consistent with local axon phenotype for IT, intratelencephalic projecting neurons. So there we plotted all the FREM3 neurons we have, and you can really see this very nice systematic gradient in morphology, depending on the location of the cell body, whether it's very close to whether it's in the upper part of layer 2, or they're systematically going deeper and deeper. Simultaneously, the apical dendrite becomes longer and longer, as of course is the apical dendrite of the two other. Now remember, this is all layer 2, 3. We have a separate study for layer 5. And we can see here, and this makes cell typing challenging because we can see both discrete groups of cells that we can clearly identify as one cell type as well as gradients. And, so here when we're just looking at FREM3 subtypes on the right, we can cluster them based on electrophysiology, morphology, or the expression of specific genes that, depending on their location, they're graded. Or you can see the by transcriptional mean in that the middle upper diagram. We project the transcriptional space, 2,000 genes there onto UMAP, and again you can see this gradation. And so were you to sample discretely from the upper and just the lower part, you would say, oh yeah, they're two different cell types. But if you sample densely enough, in this case, you can see there's sort of this continuum. And as I said, this makes this clustering challenging and somewhat partially in the eye of the beholder. It's a landscape. It's a very complicated, very high dimensional landscape, right? Ultimately, it's 20,000 dimensions, all the genes that are expressed. And you can find all sorts of patterns in there. But here, these properties very nicely correlate with the electrophysiology, with the morphology, and which is gene expression for this particular FREM3 subtypes that, in our hands at least, constitutes the majority of our 385 cells. And so, for instance, if you hear like me a lot about consciousness and we profoundly affect our consciousness, or you're also, like you guys at Harvard, at MH, I know they're starting a big center there on psychedelics. And of course, you probably all know there are roughly 60 different trials now of psychedelic, psilocybin, for treatment-resistant depression, for major depressive disorder, and for post-traumatic stress disorder, that these substances are now being tested in humans. We know almost nothing about the human. We have, of course, thousands of years of experience with these substances because people take them voluntarily. And we've now put people in the scanner on EEG while they take psilocybin and other serotonergic 2A receptor. 99% of what we know comes from studies in rodents, in mice, and in rats. And they're as I'll show you, they're very different cell type distribution, particularly with respect to the serotonin receptor gene family. And so this, of course, really matters. So on the left, you can see what we look at for the dominant human excitatory and inhibitory cell types that we found in our earlier study, and we plot the genes for the various, serotonin 5-HT, families, whether it's 1 or 2 or 3 or 4 or up to 7. There's 7 families with many subfamilies. And you can see, interestingly, particularly if you focus on 2A and then to a lesser extent 2C and 1A, that's believed to mediate much of the psychogenic effects, that in humans, at least, very little expressed in inhibitory GABAergic cells. But in particular in these cell types, these supragranular cell types. And then if you compare that, I mean, this is why it's important to study human material. So if you compare the gene expression between human and mouse, specifically with respect to these 1A, particularly in 2A receptor, you see all sorts of cell type differences. So yes, both human and mouse have receptors responding to 2A and 1A and 2C, but they're quite differentially distributed. And of course, that matters a great deal. All right, so in summary, we find that the transcriptomics of excitatory neurons in human layer 2-3 layers of cortex, of middle temple gyrus primarily, and some prefrontal cortex are correlated with their morphological and physiological features, that they collectively map to the intrateloencephalic subclass, in other words, the cortical or intrateloencephalic projections. That the deeper layer 3 cells have extensive complex basal dendrites that cover deep layer 3, but their apical dendrites rarely extends into layer 1. And so this supports some emerging evidence that at least in the primate brain, there's some evidence of this in non-primate, that superficial and deep parts of supragranular cortex really comprise two functionally independent information streams, with one information stream accessing information that's provided by feedback that goes heavily into layer 1, and then a second stream consisting among others, KM1, P1, and FREM3 that don't access that, that have heavily local connections that fire differently. They tend to fire in burst, and so that there are these two separate streams of information in superficial layers. This is our team. So this study came out of the lab of Adeline. Adeline and I are the senior authors. And you'll also hear Rebecca Hodge speaking in this seminar on a different, not on these cells, but on a very different study, but coming out of this similar large-scale project. As I said, it's a project that's been going on for a long time. Just because in neurosurgery, one has to have a great deal of patience. It took a long time to set up and to learn all these techniques to apply the techniques that were pioneered in rodent slices to human slices, and then doing all the pathology. And of course, all of that is made possible by the generosity of Paul Allen.